We do have back the test scores from the examination service, and I will be posting those on Icon probably this evening. I don't have a formal little presentation, as I often have for the tests, partly because I got them fairly close to the time coming in, but also because the results really were pretty good. The average is in the 80s, and most of the questions, 80 to 95% of the students got right, which I'm quite happy about. I'll explain a little bit more about how I look at each question, because I break it down and I look at every question in the exam to see how difficult that question was, that is, what fraction of the students got it right, and how good a question it was. There's a number called discrimination, which says if students got other questions right on the test, did they get this right? And that's one way of measuring whether it's a good question or not. There were a few questions in the test that were not in the 80s, but that's to be expected statistically. Looking at those 2 or 3 questions, the second best choice was often the next most common answer. I'll keep that in mind if it seems towards the end of the semester that there's any reason to tweak the grades, but I think for this test, it really came out about as well as a test is likely to come out in terms of being fair and covering the material and reasonably good chance for everybody to get it right. So I will be posting those scores. I had posted the answers on Friday. Are there any questions on the answers as they were posted that people want to ask? I did get a few emails over the weekend. Are there any other questions that people haven't had a chance to ask? Yes?
How much was the extra credit worth?
It will be worth up to 10 points. It has to be manually graded, so it'll be a little while before that's in. The bonus points are registered separately. So what you'll see on Icon is the multiple choice credit. The bonus credit will be a separate entry when it's done. And you don't need to ask about it. It'll be done as fast as we can do it. You can get up to 10 points. The way it'll be graded is there will be credit given for true facts that are relevant to the answer. Points will be taken off for untrue facts or false things, whether it's relevant or not. And it will only go down to 0. That is, if you told us 5 things that were wrong, you wouldn't get minus 5 points. You just get 0. So there's no advantage to putting down irrelevant material. But we do want to see, full answer to the question as it's asked. So that I will announce when those grades are posted. You don't need to worry about asking. We will announce that. Other questions? Okay. Are there any other questions about what I talked about at Wednesday at the lecture? All right, let's start back in on enzymes then. Okay. So we had gotten in. I think we were talking about the contributions, the different. Does this look about right in terms of where we were? Yeah. Okay. I'll just pick up on this in any case. If there's some duplication, at least it'll be fresher in your mind. So to come back to the question of how enzymes work this magic. That is, enzymes can accelerate the rate of a reaction. They can't make something happen that's not energetically possible, but they can accelerate the reaction literally millions or billions of fold. And some of the ways in which they pull that trick off include the following. And I need to fix my pointer. And one is simply proximity, making sure things are close to each other. This is one of the things that a powdered platinum can do, but it doesn't do it with nearly the finesse that an enzyme can. Orientation, that they're not just close to each other, but that they're pointing, the right way towards the next reactive thing. That's certainly something that enzymes can do generally better than inorganic catalysts can do. So those two together, bringing things close together in the right direction, can give as much as 1,000-fold acceleration of a reaction rate. By induced fit, it says change of shape with a substrate. The notion here is that the substrate itself binds to the enzyme in one configuration, but the fact of its binding induces the enzyme to twitch a bit, and now it's a slightly different configuration in which it facilitates the binding to that transition state intermediate. There's electrostatic catalysis, where the interior of the active site has fixed electric fields because of the side chains of the amino acids there, and those fixed electric fields create an environment in which the electron clouds of the reactants are going to be sort of urged in the right direction. And the importance of the fact that unless water is one of the reactants, it's kept out of that site, so you don't have the really common side reactions. If you think of how incredibly abundant water is, if that's a possible reaction, you don't want the water around because it will happen. And so this is giving an example of some of those features. Hexokinase is a very important enzyme. We'll be talking a lot more about it during glycolysis. It catalyzes this reaction here. It can take glucose or other hexoses and bind them. It'll also bind an ATP, catalyze an addition of a phosphate group up there, and release the ADP, the diphosphate, and now a phosphorylated sugar. And in the unoccupied state, hexokinase looks like this. But the binding of glucose causes it to clamp down like a clamshell around the reactants. In that process, it's going to strip off the water molecules. You remember when we talked about the structure of water, all those OH groups here on this glucose make it a very polar molecule, and so the water molecules can interact with all those alcohols. You have to strip all of those waters off if you don't want them to compete for the reaction. Okay. This catalytic triad in the active site is such a good catalytic mechanism that we see it in more than one place. This particular picture is showing you the tertiary structure, the overall packing in three-dimensional space, for three members of one particular family of proteases. This is chymotrypsin, which we've been talking about, trypsin, and elastase. And you can see here that the tertiary structures are remarkably similar. They're not quite as similar as the globins, but they're really pretty similar. Their primary sequences are not at all particularly similar at all. There are a few points of contact, but many, many more differences than similarities. But the asp-his-ser is conserved. The aspartate histidine serine is conserved in all of them. And so this tertiary structure folds around and creates that active site, bringing those side chains together in just the right configuration in each of them. The surface residues, since they don't have anything to do with either the enzyme catalysis, changing the rate reaction, or with binding a particular substrate, these surface residues out here that are exposed to water are much more divergent. There's a lot more variation in which ones are out there. If we were to look at another protease, this is from a bacterium called B. subtilis. This is another protease. You can tell by looking that this is not the same tertiary structure, just sort of flashing back and forth there for you. Very different tertiary structure. There's no detectable similarity in the primary structure. In the case of the previous family, there were some points of identity that you could pick out, but a lot of differences. In the case of subtilisin, there's no similarity there. However, you do see this catalytic triad of aspartate, histidine, serine that's been reinvented by this family of proteases. And this is an example of convergent evolution where the same solution to a problem has been reinvented more than once. And it's such a great solution to the problem that there are 5 distinct families of proteases that use this particular solution to the problem of how do you add a water molecule across a peptide bond. Questions so far? Okay. So far we've been talking about catalysis, what the side chains in that active site did to change the electronic structure so that bonds were rearranged. But enzymes do more than just make a reaction happen. They make that reaction happen for certain compounds rather than others. Certainly, enzymes as catalysts are not just remarkable for their speed that they can add to things. They certainly accelerate things amazingly. But enzymes are also amazingly specific. They can pick this one compound out of the entire universe of compounds, and only this one will react, or only these 3, or only these 5. So that's almost as amazing as the acceleration of the reaction itself. So how does this specificity come about? In addition to the side chains that are doing something to catalyze an actual reaction, there will be other aspects of the tertiary structure of the enzyme that are going to be involved with binding the rest of the substrate in the correct orientation. And let's just use the chymotrypsin and its relatives as a way to look at this. Chymotrypsin as a protease clips typically after a large hydrophobic side chain. So it's not immensely specific, but it is restricted to, a handful of residues. Trypsin, by contrast, cleaves after lysine and arginine. So these are fairly large and very positively charged side chains. And elastase cleaves after small and hydrophobic things like alanine, glycine, or valine. So they're pretty small and they're hydrophobic. So you can give these verbal descriptions of them. What does that translate into in terms of a molecule? And let's take a look at how the protein does that. Now, all of those peptides have in common what's called a sessile bond. Scissors cut things. A sessile bond is a bond that can be cut. So all of them have a sessile bond, a bond that's going to get cut by the enzyme when it does its thing. So that part is constant across the 3 of them, and it's going to be the same as-pis, seer, doohickey that's going to pull it off. But near those 3 residues, near the catalytic triad, there is a pocket that can bind the rest of the substrate. And in the case of chymotrypsin, you have a big, deep pocket that is a fairly hydrophobic pocket, and you can see how the phenylalanine side chain, which is a large hydrophobic side chain, can fit into that. Trypsin, which is going to cleave after, if we remember, lysine and arginine, which is a fairly large, positively charged side chain, still has a fairly big pocket, but it has an aspartate at the bottom with a negative charge. So the chymotrypsin had a serine, which can, just a little alcohol that can tolerate hydrophobic things. Trypsin has this negatively charged thing that's going to actually kind of hold firmly onto the lysines or the arginines. And then finally, elastase somehow or other has to pick out the small and hydrophobic ones. It has these side chains sticking into the pocket, filling it up, so there isn't room for much else to stick in there. You've got the valine and the threonine that are sort of mostly filling that in with hydrophobic-ish stuff. So you have actually 2 aspects of the active site that are going to be affecting the reaction as a whole. You've got the catalytic residues that are going to be involved with the acceleration, and then you have the residues in the substrate binding pocket that are going to contribute specificity. Chymotrypsin gives us an interesting example of something else as well, which is that many enzymes are not in their final state right away, or certainly among the proteases. If you look at the original molecule that is synthesized by the cells, the original molecule is a fairly long molecule of 245 residues called chymotrypsinogen. This ogen ending, ogen means the thing that will give rise to. So this is the thing that will give rise to chymotrypsin. Okay? And chymotrypsin needs to be cleaved at least once by another enzyme, by trypsin, in order to start the processing of chymotrypsin to the fully active, mature state. And so chymotrypsin can cut it once here and cut off this little piece there. This is a precursor form of chymotrypsin. It's a larger than the final form, and it is active as a protease. And it can cut the second spot there to release that dipeptide and have it be dismissed. then the second cut in hereabouts is going to give you the final mature form of the chymotrypsin and the other residues. So the trypsin starts the processing cascade, and then the chymotrypsin can finish its own processing. It's an autocatalytic process after it gets started. So what is needed for, what is happening when the chymotrypsinogen gets cleaved? Why is the precursor enzyme not yet active, and the processed form is active? Here is chymotrypsinogen, and you can see the little bits and pieces here that are going to get clipped off, and you can see that actually the catalytic triad is already in the correct position in the proenzyme, in the precursor enzyme. So the proenzyme, or the precursor enzyme, has a fairly appropriate tertiary structure, and that catalytic triad is in the right place in the active site. But when those parts are cleaved, it's going to give access to the specificity pocket, which helps select one side chain over another, and the oxyanion hole, which is an important part of one step of the catalytic process. Now, this is important enough because we have to digest our food if we're going to keep going. And if anybody in the room knows people with cystic fibrosis, you know that it can be a problem if you can't process your enzymes correctly. But there's an even more critical place where this matter of proteolytic processing of an enzyme takes place, and that's in what's called the clotting cascade. We, unless there's someone here who happens to have hemophilia, take it for granted that if you have a cut, that after a little bit it'll stop bleeding. It happens so easily, we take it as just sort of part of the way life is. It's actually quite amazing if you think about it. How can the body be ready to stop bleeding and yet not clot the whole body to solid mass of, human cheese or something? And so it's very important to be able to start and to stop the clotting process if we want it to work well. And here you can see what we're aiming at. This is a micrograph, a photomicrograph of a little bit of a blood clot. These orangey thingies are platelets that are going to be involved in the making that clot be firm and eventually to contract. And these white spaghetti-looking things are fibers of protein that are going to play an important role, and we'll see those in a moment. To form these fibers of fibrin, there's a precursor protein called fibrinogen. Again, we have that ogen ending. So this is a protein that will give rise to fibrin. And fibrinogen by itself is a water-soluble protein. It's in your blood already, and it's just floating around as a free-soluble protein. If it's processed by thrombin, which is a protease, it now is exposed as a fibrin monomer, and the fibrin monomer can polymerize with other fibrin molecules and make an extensive fiber of fibrin molecules. It turns out that to get to that right spot takes a lot of tweaking along the way. We mentioned the idea that thrombin can activate fibrinogen to make fibrin. But we can't be having thrombin around all the time, or we'd be clotted solid. So there's some kind of a way to control this in a way. And part of what we're going to see here is a cascade of events. And the nice thing about a cascade is that because each step of this cascade and of other cascades that we'll see is enzymatic, one molecule of this can make 10 or 100 molecules of that, and then each molecule of it can make 10 or 100 molecules of that, and you're getting amplification at every step. So one thing at the beginning might be 10,000 or a million at the end of it. So this amplification is one important aspect of the cascade. And because you're getting such a cascade locally, you can have clotting going on in one part of the body and the rest of the body being fluid and flowing and not having a problem at all. This is a picture of the thrombin enzyme. And you can see, if you look, that it has our old friends, the catalytic triad, in there. Here's the asp, his, SEER catalytic triad. So this is yet another family of proteases that's exploiting this particular mechanism. And what we can see happening is that there are multiple enzymes in this cascade that are going to have to be activated by proteolysis. So the thrombin was produced from prothrombin by an enzyme called factor 10A. A for activated. Factor 10A was created from factor 10 through either factor 9A or 7A, which is a tissue factor, which is sort of around in the tissues. 9 was itself activated either by 11 or by tissue factor 7. And 11 was also activated by thrombin. So something that can tickle thrombin a teeny bit allows the thrombin to propagate that signal through this cascade so that many molecules of thrombin are activated and then many molecules of fibrin are made. So you could have something that only triggers one thrombin to be active, and then you continue to have the cascade. So it's this kind of chain reaction that's going to make that local event a very extreme one without having to have it all over the place. Now, in general, a general principle in biology is if there's a way to turn something on, there's also a way to turn it off. Seems like common sense, but you should always end up looking for it if you see it. So if there are proteases and ways to turn on a protease cascade, it is reasonable to expect that there will be protease inhibitors. And in fact, that's the case. The body has a number of protease inhibitors. And some of these protease inhibitors, in fact, can bind to the active site, but not be substrates. They can, in some cases, bind with very high affinity to the active site and bind effectively, irreversibly, at least at very high affinity. And as long as it is in that active site, it's not, that protease is not going around chomping up anything else. And so these inhibitors can mimic the appropriate substrates well enough to then trick the active site, if you like, into letting it bind, but it's not likely to come back out again. Now, these things can make a difference. You may remember that elastase was one of the relatives of chymotrypsin. I mentioned that elastase is a tissue protease, not a digestive protease. And elastase is under control of things like the immune system and the self-repair system. So where the body is engaged in remodeling things or repairing them from damage or something like that, elastase will come in for a while and do its job. But elastase can be inhibited by alpha-1 PI for protease inhibitor. It used to be called alpha-1 antitrypsin, but there's no trypsin in the body. So it's just protease inhibitor now. And this makes a difference for the following reason. If you take a look here, what you're looking at is a light micrograph of thin sections of the lung. And this is what a good, healthy lung looks like. It's a lot of open spaces, but there's nice containers around each one, the alveoli, that allow for exchange of oxygen between the air and the capillaries. And that's how you can oxygenate all of that hemoglobin going out to the muscles. Here you can see the lungs from someone who has emphysema. And emphysema is a chronic, progressive, irreversible form of damage to the lungs. And you can see here that the overall tidy structure is breaking down, and these huge spaces that have opened up because of damage are not very effective at transferring oxygen because they're not surrounded by a nice bed of capillaries. Now, if you have a cold or something, or pneumonia or something like that, or you have some kind of temporary insult to the lungs, elastase will be called in during part of that remodeling process, and then alpha-1 antiprotease inhibitor will turn it off when the job is done. There are some people who are missing alpha-1 protease inhibitor, not a lot, but a few, and they are prone to get emphysema. It's not their certain fate, but they're much more likely to get it than someone who does have proper levels of it. The other people who are likely to get emphysema are people who smoke, and that's because alpha-1 protease inhibitor can be damaged by the byproducts in the tobacco smoke. So if you look here, this is a normal, healthy alpha-1 protease inhibitor. But if in someone who is smoking, a side chain, methionine side chain like this, can be oxidized like that, and you can see that it's taking something that would normally be fairly hydrophobic and making it polar now. And you can guess from the fact that when we looked at sickle cell hemoglobin, a single residue changed from polar to nonpolar or nonpolar to polar has the potential to make a big difference. And in fact, this little bit of damage, that one methionine damage, means that that whole molecule of protease inhibitor is effectively not going to work. So smoking mimics, in a chemical way, the genetic defect that happens in people who are missing it genetically. And that's part of the reason why emphysema and other kinds of lung damage are more prevalent in people who smoke. Another dramatic example of how a single residue can make all the difference. This kind of understanding of how enzymes go about their business and the relative role of the catalytic residues in the active sites and specificity pockets in active sites has allowed us to think in new ways about how enzymes can evolve. Because if you think about it, you know, these enzymes are amazing and beautiful objects. Golly gee, how does it come to be so good at what it does? And what we can see when we look at families of enzymes is something like this. There'll be what has been called like a scaffold, a part of the enzyme structure that's fairly constant in its overall configuration, and there will be certain key residues, like the asp-his-SEER, for example, that we see in the proteases. And this sort of scaffold can be seen as the building platform for other existing members of the family. And so here we have, say, family member X and family member Y. And I'm trying to see how well the colors come out. Well, they are shades of gray, at least on yours. So you can see here these loops coming around, and you can imagine those loops playing a role at generating specificity for the substrate, and even tweaking the catalytic activity of that site. It might be, you know, a protease. In this case, it might be some other kind of hydrolytic reaction in another case. And so you have these variable regions on this member that are different, distinct from the ones in the other family member, which if you were to stretch it out to look at their primary sequence, so here's the primary sequence all stretched out. And you can see how they share these key catalytic residues in common, but they have these other loops that are different, that are going to be part of, you know, substrate binding, tweaking the exact reaction, so on. If gene Y and gene X undergo homologous recombination, and that's something that happens all the time, it happens at meiosis all the time, you end up with a new gene. So if we look at it in a linear fashion, here's this new gene. It still has the key catalytic residues, the ones that are absolutely essential. And now this new member of the family has this orange block from X, and it has the green block from X, but it has, it's lost this gray block, and it's picked up this green block from Y and the pink block from Y. And so when you look at the tertiary structure of this novel family member, that arose just through homologous recombination, which goes on all the time. We see it happening. It's generated something new. This is something that hadn't existed before. So these loops in combination with each other are going to create a new space. And maybe that new space doesn't bind anything very well, in which case, nothing will come of it. But maybe that new space binds a substrate that's available in this environment that hasn't been really useful before, and now there's an enzyme that can take advantage of it. So this gives us a level of genetics and of structural biochemistry, a way of thinking about how novel specificities can arise. Any questions about this before I get into the next figure? Yes. How could what? That's a very good question. He asked, how often do enzymes arise from recombination? And my best answer for you is I don't know. I don't know that people have tried to look in populations to see. If you think about how many enzymes we have and how many generations there have been, you wouldn't need to have it happen very often for there to be a number of things for things to work on. But if it's happening at a low rate, it might be hard to observe it in a population in real time. Does that help answer that? Okay. Other questions? Okay. I think that it is kind of nifty that what we now know about structural biochemistry opens up things, and molecular biology opens up things that we could never have imagined before in terms of evolution. Now, this notion that I proposed in the slide ahead of us is clear enough as a notion, I would hope. But it's quite reasonable to ask, how likely is it to happen? I mean, can you at least set up a circumstance in which you can observe something like it happening? And this is an example. The paper that this is taken from is available on ICON if you want to look at it. This is an example of how someone basically asked in the lab, can we see this process happening if we set it up? And so they were looking at two enzymes, one that's a human and one that's a bacterial enzyme. And they noticed that the tertiary backbone, if you like, of these things had a similarity of structure to them. And so they spoke about this scaffold, this alpha-beta, beta-alpha scaffold. And they called it that because there's an alpha helix, beta sheet, alpha helix. So that's sort of the ground state, if you like. And comparing this scaffold to the end states, proposed that these two enzymes that are now in fairly separate organisms can be conceptually at least related to each other through changes from that scaffolding of adding loops that create the kinds of substrate binding and catalytic activities that are needed for the two final enzymes. So what they did in the laboratory was to take this enzyme and to remove the sort of squiggly bits, the stuff that make it specific, to regenerate or to mimic that undifferentiated scaffold. And so they have this scaffold to work from. And through a process in which you can generate variations in the gene of interest, so you don't have to just wait for them to happen by accident. But they're not directed. They're still random variations. They were able to generate a variety of sort of transient intermediates in this, and then by selection in there, find specific enzymes that, in fact, again, do what the native enzyme did before. So this is a kind of an ingenious way of trying to confirm in the laboratory that these processes are sufficient to generate novel specificities. So to just sort of recap it, you had two enzymes widely separated in evolutionary space, but which shared this kind of scaffold of alpha and beta helices. And through essentially random, they're accelerated, but they're random processes to generate variations that can then be selected by the kinds of selection that can happen in nature and discover again, the same kind of enzyme activity. Any questions about this? Okay. That's the last of our handouts, but it might be valuable if I go back over the transition state energies. Would people be interested in hearing that one more time? Yes? About what it is that enzymes actually do. Okay. Let me find that quickly. Okay. If we can get to the slide 5. I just want to go over this a second time because from past experience, it's something that isn't necessarily intuitive the first time you look at it. To look at what an enzyme does in energetic terms, we use this thing called a reaction coordinate. So it's a little bit of an odd graph because, is there a problem? It's a little bit of an odd graph because it doesn't have numbers at the bottom. What it has are the different states along the way in the course of a reaction. The vertical axis is a numerical axis. It's free energy. But it's hard to measure actual free energy of compounds. So what we talk about are the differences, the delta G's of different parts of the reaction pathway. And if for this particular one, we're starting with reactants AB and a second one C. In the transition state, there's this intermediate that has some configuration with A, B, and C all in the same thing. That's the transition state for this hypothetical reaction. And then the product in this case would be A by itself and B and C together. The difference in energy between the reactants and the transition state is the free energy change associated with the transition state. In addition to a transition energy for a delta G for the transition state, we can speak about a delta G for the reaction overall. And so if you have reactants at some free energy and products at a different one, if overall there's a negative free energy change, we can talk about that being a spontaneous reaction. That at some rate, things will get over that free energy hill. So there's a transition state between the reactants and the products, and there's some kind of a free energy difference associated with that. So at some rate, they'll get over that hill and over to products. That means it will be spontaneous, but that by itself doesn't tell us the rate. The delta G of the reaction cannot tell us the overall rate of the reaction. It can only tell us the equilibrium distribution, how spontaneous it is, how far it will go to completion when it's been catalyzed. If, by contrast, you have reactants that are at a lower free energy than the product, so there's a positive delta G, this one will not go spontaneously. But it can be coupled to something else that would drive it. And we talked about that back at the beginning. So the magic of catalysis consists entirely in finding some other way to get through the transition state. If this is the delta G associated with the uncatalyzed reaction and the uncatalyzed transition state, if there's some way that the catalyst can find a lower energy way of getting there, of getting to a transition state that would still produce the same products, I need to fix my thing here. If you can lower the height of that transition state, then you can accelerate the rate of the reaction. The delta G for the reaction overall is going to be the same. The difference between the reactants and the products has not been touched. Only the rate at which you get from one to the other. Okay. Are there other aspects of enzymes that people wanted me to go over again? I'll see you Wednesday. Hi.
I just wanted to let you know on Friday I'm not going to be here.
Okay. was